Enzymes Journal Structure. In this lecture, we will study about the journal structure of enzymes and the role of cofactors and coenzymes. Let us start with the introduction. Enzymes are proteins that act as a biological catalyst in living organisms. These enzymes accelerate the rate of chemical reactions. All the biochemical reactions that occur within living organisms are regulated by enzymes. The molecules upon which the enzymes may act are called substrates, and they convert these substrates into product. A large protein enzyme molecule is composed of one or more amino acid chain, which is called polypeptide chains. The amino acid sequence in a polypeptide chain determines the characteristic folding patterns of the protein structure, which is essential to enzyme specificity. If the enzyme is subjected to changes such as fluctuation in temperature or pH, the protein structure may lose its integrity. That means it becomes denatured and it also loses its enzymatic ability. Structure of enzyme. There are two types of enzyme: simple enzyme, conjugated enzyme. The simple enzyme consists of only proteins, while the conjugated enzyme, which is also known as holoenzyme, consists of protein part and non-protein part. The protein part of holoenzyme is called apoenzyme, and the non-protein part may be the cofactor, coenzyme, or it may be a prosthetic group. Holoenzyme. The apoenzyme is the protein part of the conjugated enzyme, which is the holoenzyme, and the coenzyme or cofactor is the non-protein part. The combination of the apoenzyme with the cofactor results in the formation of a functional enzyme, and this functional enzyme is called the holoenzyme. Apoenzyme. The apoenzyme behaves as a true catalyst during enzymatic reactions. It takes part in the reactions, accelerates the rate of reaction. However, after the reaction is completed, it always returns back to its original physicochemical configuration. The coenzyme part, however, may change after the reaction, and another reaction is required to revert it back. Cofactor. The cofactor is a non-protein chemical compound or a metallic ion that is required for an enzyme's activity as a catalyst. Cofactor can be considered as helper molecules that assist in biochemical transformations. The cofactor may be either coenzyme, which is an organic molecule such as vitamin, or an inorganic metal ion. Some enzymes require both the coenzyme as well as inorganic metal ion. Prosthetic group. The prosthetic group is a tightly bound specific non-polypeptide unit which is required for the biological functioning of some of the proteins. The prosthetic group may be organic compounds such as FAD, PLP, or it may be an inorganic metal ion. Some inorganic element as a cofactor of enzymes to which they are associated. For example, copper act as a cofactor in enzyme cytochrome oxidase. Potassium act as a cofactor in enzyme pyruvate kinase and propionyl coenzyme A carboxylase. Molybdenum act as a cofactor in nitrate reductase. Nickel act as a cofactor in urease. Selenium act as a cofactor in glutathione peroxidase. Magnesium act as a cofactor in enzyme hexokinase, pyruvate kinase and glucose 6-phosphatase. Manganese act as a cofactor in arginase. Iron as Fe+2 or Fe+3 as a cofactor in cytochrome oxidase, catalase and peroxidase. Zinc act as a cofactor in carbonic anhydrase, alcohol dehydrogenase and carboxypeptidase A and B. Now the various coenzymes and their sources and the groups which they are transferred are presented in this table. The coenzyme A, which is obtained from vitamin B5 pantothenic acid, transfer the acyl group. Coenzyme B12, also known as 5-deoxy-adenosylcobalamin, obtained from vitamin B12, transfer hydrogen atom and alkyl group. FAD, flavin adenine dinucleotide, obtained from riboflavin vitamin B2, transfer electrons. Similarly, NAD plus nicotinamide adenine dinucleotide, obtained from niacin vitamin B3, transfer hydrogen ions. Pyridoxal phosphate transfer amino group and it is obtained from vitamin B6. Similarly, tetrahydrofolate obtained from folate vitamin B9 transfer single carbon group. Similarly, thiamine pyrophosphate TPP transfer aldehyde group and it is obtained from vitamin B1 thiamine. And the last is biocytin, which transfer carbon dioxide group and it is obtained from vitamin B7 biotin. And these coenzyme plays a very important role in the transfer of the group and also plays a very important role in the activity of enzyme. Active site. The active site is the region of enzyme where the substrate molecule binds and undergoes a chemical reaction. As we see in this picture, this is the substrate and this is the enzyme and this is the active site of the enzyme. The substrate molecule binds to this active site. The active site consists of amino acid residues that form temporary bonding with the substrate and residues that catalyze a reaction of that substrate means catalytic site. The active site may be either at the surface, for example in chymotrypsin, or it may be in a cleft, example ribonuclease in the apoenzyme part. Active site is made up of few amino acids and it has a certain degree of flexibility in its structural organization so that it can accommodate substrate molecules. The tertiary structure of the enzyme protein is folded in such a way so as to create an active site that has the correct molecular dimension and appropriate topology to accommodate and bind with the specific substrate. monomeric and oligomeric enzymes. Enzymes exist either as a single unit, means they consist of only single polypeptide chain, are known as monomeric enzyme, or when enzymes are the aggregates of several subunits, means they consist of several polypeptide chain, they are known as oligomeric enzyme. Each subunit of oligomeric enzyme have an active center where a substrate molecule bind during the enzymatic reaction. Thus, the oligomeric enzymes are different in their kinetic behavior than the monomeric enzyme. Allosteric site. The allosteric site of an enzyme is different from that of the active site. The allosteric site is a site of the enzymes that allows molecule to either activate or inhibit the enzyme activity. This site is different from that of the active site of the enzyme. On the active site, the substrate molecule bind, whereas on the allosteric site, effector molecule bind. When allosteric activators bind to the allosteric site, the enzyme bind the substrate better and the reactions becomes faster. This is the active site of the enzyme where the substrate molecule bind, while this is the allosteric site of the enzyme where the activator or effector molecules bind. The effector may be positive effector molecule or effector may be the negative effector. When a positive effector binds to the allosteric site, it gives the catalytic site of the enzyme a conformation which is more suitable for the substrate to bind to the active site. But when a negative effector binds to the allosteric site of the enzyme, it gives the catalytic site of the enzyme a conformation which is not suitable for the binding of the substrate to the active site. As a result, this allosteric inhibitor inhibits the binding of the substrate. Enzyme shows absolute specificity. Enzymes shows absolute specificity in terms of substrate on which they act and in the types of reaction being performed. Some enzymes shows absolute specificity for the substrate, while other enzymes have a broader specificity and they accept several different analogues of specific substrate. For example, glucokinase is specific for glucose only. On the other hand, the enzyme hexokinase catalyzes the phosphorylation of glucose, mannose, fructose, glucosamine and 2-deoxyglucose, though the reaction rate is not the same. The specificity of the enzyme is due to its three-dimensional structure and due to its active site. For further reading, I would like to suggest these two books of biochemistry. One is the book of biochemistry by S.N. Gupta. Another is the Fundamental of Biochemistry by Jain and Jain. Thank you for watching.